The tau kinase was first identified in plants as a gene that is involved in some aspect of flower development, and we found the homologue in humans. Initially, our work was on the function of this protein kinase in the DNA damage response, but then we expanded, to aspect of cancer, and particularly prostate cancer. I should say that it's important to know that if you go to the ULCAM database that essentially correlates the expression of genes with disease progression, then tau-like kinase is a better prognostic marker for not very advanced disease. So if you have a Gleason score, which is essentially what the pathologists use to classify the danger, let's say, of the disease, then when TLK1 is elevated and the Gleason score is not particularly high, say 6 or 7, then it's a bad prognosis. So it's important. There is genetic evidence that it's important. What is the impact in the field? Maybe I'll go to how it works. So the main research in our lab is understanding the function of this protein kinase in prostate cancer progression. So it works particularly on two pathways that we identify. One is the HIPPO pathway, where TLK1 phosphorylates the transcriptional coactivator YAP, which is the last downstream part of the pathway. It's really what controls the expression of some genes that are involved in both drug resistance and CRPC progression. And TLK1 phosphorylates a site that is important for the stability of the protein. Normally, YAP is an unstable protein. TLK1 phosphorylates and stabilizes it. So essentially, it increases its expression and then essentially participate in the reprogramming of the cancer cells to more aggressive phenotype, drug resistance, CRPC progression. The other pathway that we identify is that it phosphorylates another protein kinase that is called MK5, or MAP kinase activated protein kinase 5. This was identified earlier as a protein kinase involved in invasion, metastasis, motility, particularly motility. And so TLK1, we found that it phosphorylates a site on MK5 that leads to its activation, and this essentially renders the cells more motile. And also more invasive. It affects also the expression of some metalloproteases. So it also, so it's more motile and also more invasive through tissue, through the stromal layers. So these are the two main pathway that we worked on. So TLK1 is involved essentially in cancer drug resistance because of this, and prostate cancer progression, CRPC progression, because of these two factors. I should say that YAP can convert androgen-dependent cancer cells like lung cap to androgen-independent. So it integrates the androgen receptor signaling of genes so that they become more active even when there is low androgen. So it amplifies the androgen signal even when there is low concentration of androgen. So yes, we had initially done a brute force drug screens to find inhibitors of TLK1 and TLK2. And we had identified several in the class of these phenothiazine antipsychotics. So there is a class of these drugs, and taurizazine was the most powerful one. So it was a good inhibitor for TLK1, but these are repurposed drugs. So their main function is actually to be antidopamine antidopaminergic drugs. So the problems with these drugs is they essentially make the person lethargic. So it's not really pleasant. And two, they actually have a more serious effect in that they can cause cardiac arrhythmia. Again, the heart is affected by the autonomous dopaminergic system. So so we designed a drug that had much less affinity for the dopamine receptor, still in this class of phenothiazine, and it does work well. So if you combine J54 with antiandrogen in the LANCAP model of xenograft, it not only stops the cancer cells from progressing into the androgen independent phase, it actually shrinks the tumor. And the mice are fine. They're not lethargic like for the couple of hours that after the injection, the administration of thyroidazine. So it looks like it's much safer and it doesn't have this bad effects. Essentially these drugs have these antipsychotic drugs. Therapy is a new way of treating early the early treatment of prostate cancer. So if the physician feels that the prostate cancer is fairly advanced, like PSA level is particularly high, then before surgery or radiation, they will initiate antiandrogen therapy to both shrink the prostate tumors and also to essentially start, if they suspect that the prostate tumor may already be partly refractory to antiandrogens, then this early treatment may buy some time. The question really that I have is whether this 50 years of treatment that we're doing with antiandrogens, so there is just a uniform type of treatment for prostate cancer, and that is the ADT, the androgen deprivation therapy, and antiandrogens. And sure, prostate cancer was found to be androgen dependent, but we have not really made. We continue to make a better mousetrap, and we most of the work in the pharmaceutical industry is to make better and better antiandrogens that will work for longer on the androgen receptor. And my question is whether it is really the only option to go. For example, new research that we're doing now is to see if we can make better use of carboplatin or essentially cisplatin, which is not the therapy of choice right now for prostate cancer, mostly because of the dominance of the anti-androgen deprivation therapy. And two, because, it does have some renal toxicity, so it's not the first line of choice. But if you combine, they found, in several studies, if you combine lower doses of cisplatin with inhibitors of the DNA repair pathway, of which TLK1 is a main component, then you can use the treatment much better. And the question is, maybe it's time to come out of the dominance of the anti-androgen therapy and start to think about other ways of treating prostate cancer with different ways. Anti-androgen also have some bad side effect for men. So it may not, maybe it's time to break the dogma and start thinking about other things rather than just thinking about new ways of overcoming CRPC, which may be not possible. So it's just cancer cells are not that's stupid. They reprogram and they find ways of recovering from the androgen deprivation therapy. Well, I think this is again, it's a reinforcement of what I was saying before. I think they're having much success now, at least for longer survival times, with the use of PARP inhibitor in combination with antiandrogens to have a better result and delay the death essentially from CRPC. I should say how this works because it actually impacts also our research. So antiandrogen, at least in the one model that is most studied, which is the lung cap cells, cause a G1 arrest by triggering the DNA damage response. So and the cells essentially become quiescent until they reprogram to the new conditions, and then they become androgen independent, which is essentially CRPC. So the PARP inhibitors essentially are essentially inhibitors of several DNA repair pathways, particularly the nucleotide excision and the base excision repair pathways. And essentially what the cancer cells, the prostate cancer cells do is they arrest in G1, and essentially they buy time to increase the DNA damage that is caused by essentially incompletely replicated strands of DNA, so that they don't enter mitosis with a catastrophe, essentially, because they have incompletely replicated DNA that's going to cause massive amount of damage when the chromosome segregate into daughter cells. So the PARP inhibitors essentially are doing that. Essentially, they inhibit one pathway of the DNA repair pathway. So are the PARP inhibitors the only thing that can be done? There are other drugs that affect other DNA repair pathways. And for example, TLK1 and therefore J54, the inhibitor of TLK1, are involved in the homologous recombination DNA repair pathway. So I think the future I think is basically attacking this DNA damage response and repair pathways that are a component of the anti-androgen, the androgen deprivation system of prostate cancer cells. And I think that's going to be probably the main advance in the next few years. Yes, so recently we found that TLK1 is an important factor in homologous recombination repair by phosphorylating the key recombinase cooperating factor that is called RAF54. It's essentially it's a strand displacement ATPase that essentially allow the loading of RAF51, which is the main recombinase. So I think it goes to what I was saying before. I think if you can inhibit DNA repair pathways, you essentially and also bypass the DDR, the DNA damage response, then essentially you end up with cells that have progressed into mitosis with unrepaired DNA damage, and that is going to cause essentially massive apoptosis. So, rather than stopping cancer cells with antiandrogen alone, because that's just it's a band-aid. You have not really killed the cancer; you just stopped it for a little bit. So the best option is actually to try to destroy it, and essentially, so the bypassing the DNA damage response, forcing the cells into continuing catastrophic replication, and also preventing the repair of the damage, I think it's going to be really the research in the future, and this is really what my lab is working on. Essentially, we are trying to understand, and we made significant progress in understanding how TLK1 is involved in both the DNA damage response and in the DNA repair pathway, particularly the homologous recombination repair pathway.